Hello, everybody, and welcome to Central Saint Martin's Digital Innovation Season. As you know, we have talks every Tuesday until the end of May, and the theme of this year's season is the intersection between technology and biology. In addition to the public talks, we also have a series of 27 workshops for our students to learn digital skills such as machine learning, computational arts, or robotics. Today's topic is biorobotics and artificial life forms. We will look at how robotics draws inspiration from living systems, not only in their shapes and movements, but also how they grow, how they self-assemble, how they learn, adapt, and evolve. And the researchers that we are hosting today are looking beyond mechanics and into cellular biology. They use living cells and artificial cells. They grow them in the lab and they can program them, hence turning them into robots. This means that we can harness the communications between cells, the chemical signals between these highly complex biological systems in order to perform movements or tasks. And perhaps in the future, we won't need any more metals and plastics in our electronic devices, and we could move fully to organic living materials. This approach also challenges the definition of life. Life is traditionally defined by three characteristics: the capacity to grow and to self-replicate, to feed on some sort of energy, and to have a metabolism or an operating system of chemicals. So today we will be looking at different types of artificial life. And I'm extremely delighted to welcome three pioneers in biorobotics, Dr. Park, Dr. Cheskova, and Dr. Sina Payne. They will present one by one, and we will take questions at the end of the event. So please do write your questions in the chat on YouTube. So first, I'm going to introduce Dr. Park, who is an assistant professor at Georgia Tech. He's based in Atlanta in the USA, so it's very early for him. It's 7 a.m., 7:30 a.m. In Georgia Tech, he leads the Biohazard System lab, and he has developed a biohybrid stingray robot a few years ago using stem cells. At the time, it was rat cells. And more recently, he's developed a biohybrid fish using human cells, human heart cells that are contract and are controlled by light signals. So his field of research is the study of the structures of biological systems which are behind the production of physical behaviors. I'm going to hand over to you, Dr. Park. You can start sharing your screen. You're on mute.
Thank you for introducing me. Today, I'm going to talk about the biohazard system. So people start to think about the integrating the living cell and tissue into the artificial synthetic platform. That is the definition of the biohazard system. So probably most of the people not familiar with the word, the biohazard, maybe first time is the biohazard system today. But you can easily find the scientific movies, scientific fiction. For example, it's the Replication 2010 movie. So this movie is showing that people start in the future. People start to integrating the living cell into the metallic structure and to build the artificial organ. So people simply replace the damaged the tissue using this kind of the metallic-based biohybrid system. And then 2017 movie, Blade Runner, probably most of people know this movie. Here is a more advanced form of the biohybrid system, the kind of humanoid robot. The movie is to say that it's replicants. So basically, this humanoid robot is entering the biohybrid system more soft and living that biomaterial form, and to produce the more like a human-like behavior. And then recently, the people start to think about the artificial meat. So it's people start to think about the artificial culture's meat, then this is also is one of the biohybrid system. So because the entering the living muscle tissue into the living muscle cell into the plant-based the synthetic the biomaterial scaffolds. So as you can see, the living biohybrid system will be beyond the movie. So in the future, this system will be is part of our life. So in currently, there is the industry, the research fields, the industry. So it's the first thing is the drug screening, the system. Many companies already working on this kind of the biohybrid system. So it's basically instead of using the animals, we can use the human tissue as the drug screening platform. And then many researchers currently working on the artificial muscle tissue or the muscle patch. So that will be one of the biohybrid system form. And then is the cultures meat, as I described it before, is several company already, 7 or 8 company already out there to trying to build the artificial meat. So it's the designing the biohybrid system is quite similar with the conventional mechanical system design. So basically, as you can see, the mechanical system or the robotic electronics, the system, as you can see, it has the control unit, basically mechanical, the leverage here, or the CPU based the electronics FPGA here, and then it's functionally connected the control unit into the functional unit, and then trying to build the system level structure, and then make them produce the desired behavior. So likewise, the biohybrid system, basically, we're trying to integrate the regulatory mechanisms, which is the biological cell and tissue or protein. And then in order to regulate the functional network of the living cell and tissue. And then ultimately, we control the external stimulus to control the regulatory mechanism and basically to induce the desired emergence behavior of the biological system. So it's the three importance, the design, the concept we have to think about. First one is scale, is basically we cannot build the really complex the network of the components in the natural form. So we have to think about what is the minimum functional unit needs to replicate the targeted function. That is kind of we have to think about this kind of process in the design process. And also we have to think about how we control, how we measure it. So that is, we usually use the biotechnology technique to measure and control this kind of the living system. So it's so far is I built the 4 different systems. And today really focus on the 2 systems, which is the human biohybrid fish and tissue engine stingray published in 2016. So first thing I'm going to focus on the stingray. At that time, we simply think that we trying to build the heart, build the artificial heart. But at the time, it's really difficult to build the 3-dimensional structure of the heart and then 3-dimensional motion. So we start to think about this more simpler, make the problem is simple, make the simple problem. So we identify the 2 functional structure. So first one is we think that is the heart has the muscle or its unique muscle arrangements. As you can see, the heart has connected, the heart cell is connected to each other, form the tissue, and then each tissue is more like a laminar form, and that is wrapped around the cavity of the heart. So it's when they muscle contract, then it's kind of they squeeze out the muscle blood from the cavity. And then they have also is really the unique conduction system. So it's basically the muscle activation, the rhythm is generating from the sinoatrial nodes that is propagated through the muscle tissue, and then it's induced the sequential muscle activation that is induced the ventricular torsional movements of the heart. That increase the muscle activity and muscle efficiency. So we want to try to build this kind of the 2 major components of the heart into the simpler form. So we look around the several different biological model. We think that is the stingray is one of the simplest form because they have the really planar body structure compared to the 3D, the structure of the heart. So, but it has to contain the unique muscle arrangements, and also has the conduction system. We think that we build this kind of the muscle arrangement of the conduction system of the stingray, then we can measure the swimming speed of this biohybrid stingray. Then maybe we can learn the meaning of this kind of the muscle activation of the heart. So as I say, is that we need the regulatory mechanism control the biohazard system. At that time, we think that is optogenetics, basically the genetically engineer the muscle tissue with express the light-sensitive protein, which is the channelopsin here, and basically genetically engineer the muscle cells and make them muscle is light sensitive, and then we can induce using the light control the muscle movements. Basically, the functional network is special muscle activation that is induced the undulatory motion of the stingray. So it's a shining light here, and then there is a muscle pattern is here, and then that muscle activation wave is propagated through the muscle tissue, unique muscle tissue that is induced the sequential muscle activation and sequential muscle contraction that is induced the undulatory fin motion of the stingray. So to do that we really copy all of the muscle structure from the stingray. And as you can see, the stingray, our biohybrid fish, the stingray is 10 times smaller than the juvenile stingray, but the muscle architecture is quite similar with the stingray. And then it's using the optogenetics, as I said before, so we can using the light to stimulate the muscle activation in the front muscle first, And then that is muscle activation pattern is propagate through the muscle tissue, as you can see, and then induce the undulatory motion. Finally, you can see this kind of the movie. So here is the muscle, the light here, and that is the induced muscle activation, and then it's swimming towards to the light stimulation. So we can say that this kind of the fish is kind of the robotic system. So what we learned from this kind of project is the deformation pattern is really important to improve the performance of the stingray. So as you can see, this is the stingray, the live stingray, and then this is our biohazard fish. You can see that it's compared the stingray and the biohazard stingray. We actually replicate the deformation pattern of the stingray. So that is improve the muscle move, the swimming speed of the stingray. And then one of the great thing is that this stingray is actually we can control the left and right muscle. So we can make the turn using the light stimulation. For example, is that if we stimulate the low frequency of the left muscle and the high frequency of the right stimulation in the right muscle, then it's the stingray is moved towards to the left. So using this kind of technique, actually using the light stimulation, we can make them swim through the obstacle course. You can see this kind of the movie. So that's why we really start to say that this biohazard system is a robotic system, basically, but it's definitely a simpler form of the just conventional mechanical, the robotic system. So next project is based on this kind of the achievements of the 2016. So, but it's at the time is as you can previous movie, we have to stimulate continuously stimulate the biohazard system. So we start to think we can integrating the intrinsic regulatory mechanism instead of the extrinsic external regulatory mechanism. So first thing is we think that is the mechano-electrical coupling of the heart is probably we can use the regulatory mechanism which we don't need the external stimulation. So heart has the unique mechano-electrical coupling system. So basically the heart dynamically responds to physiological demands through this kind of the mechano-electrical coupling. They have the stretch activate the mechanosensitive protein. So it's the activation of the stretch activate the mechanosensitive protein to induce the muscle activation. Another mechanism is the intrinsic regulatory mechanism in cardiac pacemaker cell, and then it's pacemaker's tissue. But as you can see, heart has the sinoatrial node. Here is the sinoatrial node. They initiate the muscle activation rhythm of the heart. And they have the unique structure, you can see. They have the isolate this kind of pacemaker cell from the muscle tissue. They has a lot of the connective tissue surrounded by this kind of the pacemaker tissue. So it's easily activate the intrinsic rhythm out of the muscle. So we kind of the copy to the stretch activated channel of the heart, and then it's the unique structure of the sinoatrial node. We integrate into this kind of fish platform. As you can see, the stretch to control the stretch integrate the stretch activate the protein. So what we do is that we make the two layers of the muscle. One is left and the other side muscle is right side muscle, and they connected each other into like a soft gelatin the material. And then it's when the right side muscle activate, and then that is the left side muscle is stretches. So it's a stretch activate, the protein is activate. So that is induce the muscle activation in the right side. And another one is the copy the structure of the sinoatrial nodes. So we isolate the kind of the muscle tissue out of the big muscle tissue. So it's that it easily initiate the intrinsic muscle rhythm from the sinoatrial nodes, from the we call the G node. So as you can see, the fish has the double layer of the muscle. So this is bottom layer, and this is left side, and this is right side. And then each one has express the light sensitive, different type of the light sensitive protein. So it's the left side muscle is blue light activate, right side muscle is the right side, left light activate. To do that is basically we can, sorry. So as you can see, we can control the muscle. First, we control the muscle is optogenetically. So basically, red light is control the left side muscle, and blue light is right side muscle. And you can see that it's alternating the red and blue light to induce the muscle contraction, and then make them swim towards the stimulation. And then is after that is we testing the intrinsic control. So first thing is we see that is as we design the stretch activated channel platform. So you can see that it's without any stimulation, the stingray, the biohazard fish is contract automatically is contract, you can see. So it's without any stimulation. So it's left and right muscle, so it's the left muscle contract. Then it's induced the stretch activate channel that is induced the right side muscle activation, and so on. And then is the G nodes, the structure we integrating here. So you can see this is a propagation of the muscle activation. And then you can see that it's muscle activation induced from the G node, which is isolates the muscle tissue out of the big muscle. So that's why this kind of muscle has muscle has the unique feedback control loop. You can see as you usually find any mechanical robotic system. So we have the mechanosensor, stretch activate the ion channel, and G node, which is the pacemaker like a tissue that is regulate the muscle activation, that is kind of fit into the closed loop. So you can see that this is the first system to integrate in the closed loop system. And then it's why we can control the optogenetic external stimulation to control this muscle movements. So that's why we can make them swim. It's kind of much faster than any other biohazard system. As you can see, this is a real-time movie. The fish is move kind of more than 1 centimeter per second, and that is it's much faster than any other biohazard device so far. And then so the efficiency also we can improve. So it's using this kind of intrinsic mechanism. So we demonstrate that our biohazard system performance is similar like the small size of the natural fish. And then one of the good things is this intrinsically swimming the biohazard fish is we can extend the longevity of the fish. You can see that this is the 108 days the fish, So they swim by themselves without any stimulation, so they can survive much longer than any other the biohazard system. So that is the last slide. So it's the continuously we focus on this kind of the studies. So we're trying to build the more bigger size of the biohazard system. Also, it's more like an adaptive fish biohazard system to the response the various the environmental stimuli, and also is we focus on the make them is more efficiency, the improve the efficiency of the biohazard systems currently. So that is the last my slide. Thank you.
Thank you very much, Dr. Park. It was truly fascinating. Fascinating to see how you combine engineering and biology knowledge to develop these truly cutting edge technologies. So now we're going to move to Dr. Českova's presentation. So we're moving from living cells, stem cells, into artificial cells. So Dr. Českova is an associate professor of chemical engineering at the University of Chemistry and Technology in Prague. She leads the droplets lab at this university, and she studies how living cells move, and she has also researched the origins of robotics, and in particular where the word robot comes from. And so she has developed a concept called droplets as liquid robots, for which she has received many awards. And this concept is based on protocells, which are artificial cells made from chemicals which behave like living cells, and they are believed to have had a role in the origins of life. So I'm going to hand over to Dr. Českova.
Good afternoon, or evening, or morning. Do you see my shared screen?
Yes.
Yeah. So the title of my talk that I have prepared for today is Liquid Robots, and I will speak about robots without sheet metal and wheels. And I use this sentence from Karel Čapek, and I will tell you more why I use this title and what I am working on. And first of all, I would like to start with this. If we say a robot, each of you have in your mind what robot is. If you Google the images for robots, you can see different robots, from industrial robots through humanoids to kitchen robots. But never you can find something like this. And do you think that these guys are robots? Actually, yes. And they are the first robots, because these two persons are actors from the theater play Rozum's Universal Robots. And this play was written by Czech writer Karel Čapek 100 years ago. And this story, maybe some of you know this RUR, Rozum's Universal Robot, so I'm sorry that I will repeat it for those that they don't know where the word robot comes from. So in this play was the mad scientist, his name was Rozum, and he decided to create artificial human beings. And these robots were from flesh and bones. They looked really like human beings, and not they have not contained metal or some electromechanical components as robots today. And actually, in this play, the robots were described that they are created from the living matter, and Čapek used the sentence that this living matter was kind of colloidal jelly that even a dog wouldn't eat. So in principle, the first robots were closer to our research, where we study the liquid robots or chemical robots, It means the robots that are from chemical and soft substances in comparison with the other robots, which are from metal and electromechanical parts. And as I mentioned, this play was written in 1920 and the premiere was in 1921. So last years we had the anniversaries of the play and the book, and we celebrated the centenary of the word robot. And I see that there is the connection between this old theater play, RUR, and the current research, artificial life. Because in the play, it is described that this mad scientist Rozum has discovered another way how to create life. And the researchers in artificial life field, they have similar objectives. And in 1987, there was the first conference on artificial life. And the founder of the field of artificial life, Christopher Langton, he said that biology studies the life as we know it, but artificial life studies the life as it could be. So the A-lifers, the artificial life researchers, they focus on systems that are not living, but they look like living systems. So yeah, so the research, the artificial life research can be divided into three main parts. One of them is soft A-life. It focuses on software, and the researchers study, for example, artificial neural network, cellular automata, game of life, and these things. The hard A-life is more related to hardware, so the typical robots that you know. But I belong to the part which focuses on that artificial life. It is related to chemical or biochemical systems, and the idea is to create in laboratory something which looks like a living system, or at least behave in some way as living systems. And if we take the comparison to Čapek's idea, where he used the word robot for artificial human beings, we can use the word liquid robot for artificial cells. Because robots, they look like humans, but they don't have all attributes of humans. They don't have feelings. They are not able to do everything as humans do. And in similar way, I use the term liquid robot for something which looks like a living cell, but it does not have all attributes of life. This slide I am using for the comparison of living systems and our research on droplets. The movie that you can see, that yellow one, is my old movie from my diploma, where I studied the living system. It was Dictyostelium discoideum. And this is very interesting model biological system where we can observe a lot of biological phenomena. We can see how single cell moves, and the cells are able to perform chemotaxis. And in the same way, I study the single behavior of, or the behavior of single droplet. In this biological system, we can study the behavior of population of cells, and in the same way, I also focused on the swarms of droplets. So this is the comparison that we take the inspiration from nature to study the droplets that I call liquid robots. And here it is again summarized what we can observe in the system that I study. I will show you the examples in the next slides. But what I want to mention here is that artificial life is related to biology, because we can compare the biological systems to our artificial systems, which are completely chemical without any living part in our case. And for our research, because I am from chemical engineering department, we use also the approaches like to study the droplets from the point of view how the physicochemical properties affect the behavior. So we use the tools of biologists to use microscopy to evaluate the speed of movement, but also we use the tools of chemical engineers. And now I will tell you more about our research. In the title of my talk is Liquid Robots, so I will speak about liquid robots in the forms of droplets. There are many droplet systems. You can check the literature and you can find different droplet systems with interesting behavior. But in my case, I studied the chemical system, which is composed of decanol droplets. Decanol is the alcohol with 10 carbons in the chain, and it is not miscible with water. It doesn't have the color, so we use the red dye to be able to observe the droplets. The droplets are in the aqueous solution of sodium decanoate. Sodium decanoate is kind of surfactant. And in some cases, we use the salt in our experiments. In some experiments not, but in some it is important part of the experiments. And now I will summarize what we are able to study and what we observed. This was one of the first interesting behavior. In nature, the cells or small organisms, they are able to follow the gradients of chemical substances. If they feel something which is interesting for them, like food, they are able to run towards these chemical substances. It is positive chemotaxis, or they can run away from some other substances, which is a negative chemotaxis. And in our case, we have observed that if we have the Petri dish with aqueous solution of sodium decanoate, and we have the decanol droplet, and if we add salt, we can observe similar behavior as in the case of living cells. I'm sorry, I don't know why the movie is not working, but you can imagine that this living cell moves towards the addition of chemoattractants, and in similar way these decanol droplets follow the gradients of salt, and we call this artificial chemotaxis. This was simple experiment in Petri dish. Then we did more complicated experiments, for example, in the maze. Maybe you have seen this because this movie has over 300,000 of views already on YouTube. So in this experiment, again, we had the source of salt in this droplet, and this decanol droplet was able to follow the gradient of salt and solve the maze. We also did experiments with this artificial chemotaxis, where we used the droplets as the transporters of small objects. And we started to call it chemotaxi. It is the taxi which is based completely on chemical signals. In the same way as the taxi can transport some passengers, in the same way our decanol droplets are able to transport some small objects. They are the limitations with weight and size and so on. And these experiments that we did with chemotaxis, they are nicely observable in the real time. The experiment takes like 5 minutes or more, so still it's possible to wait and watch it. But when you keep the experiment in the laboratory, the water starts to evaporate from the aqueous solution, and the decanol droplets, the red droplets, they start to change their shape. And we started to focus also on this phenomenon, because it is very interesting. If you have the decanol droplet, it is round shaped at the beginning of the experiment, and later on the water starts to evaporate from the aqueous solution. It means that the concentrations of the surfactant in the aqueous solution start to change, and the decanol droplet creates multi-armed objects. And it is interesting to observe it by eyes, but also under the microscope, we can observe the growth of myelin figures. So this is very nice experiment, which I think looks like it is something living, but it is only chemistry, again, only decanol, aqueous solution of sodium decanoate and salt, and we can observe something which looks like a living system. And if we compare these droplets which are able to change the shape, it is very similar to living cells, because in nature rarely you can find perfectly round-shaped droplet or round-shaped cell. Usually they have some appendages and different shapes. So in the same way, if we want to create artificial cells, we should focus on their shape and not to play only with liposomes or droplets that have a round shape. And up to now, we focused on single droplet. But what interested us was also to check if we have the population of droplets, if we have more droplets, if they are able somehow interact. Because in nature, we know that the swarming behavior is very common. Even in the robotics field, we know the swarm robotics, and people are, they study the swarms of robots. And so we decided to check if also droplets have some interesting behavior. And we have found that they have interesting behavior, because if we performed a lot of experiments where we were able to change the number of droplets, size of the system, concentrations of surfactant and so on. But in this case, I show you the experiment where we had the glass slide with the size of 24 times 24 millimeters, and 1 millimeter of the milliliter of aqueous solution of sodium decanoate, and then decanol droplets, 1 microliter each. And what we can see is that around 40 minutes from the beginning of the experiment, the droplets are, I say, dancing, and then they create the cluster, and this cluster with the hexagonal structure is stable for several hours. And after 3 hours, the cluster disintegrates, and the droplets separate again, and they start to change their shape. So this is interesting phenomenon, and now we know how the composition of the system affects this behavior, but still we are not sure why it happens. And another interesting property of decanol droplets that can be used for liquid robotics or in robotics in general is this rotation. Because if we have the droplet, for example, in this experiment, it is in the middle of the glass slide. And if we have some object on it, it can turn the object, and the droplets, they are able to self-rotate independence on the size of the system, on the concentrations and so on, up to 6 hours or more. So it is very interesting. And maybe if we imagine some mechanical machine where we have these wheels, maybe in the future we can use the droplets instead of them to be able to control the movement of robots thanks to these rotations of droplets inside. So now I have shown you a lot of videos where we had the chemical system and it looked like there is something maybe alive. And here I would like to summarize everything with the question how we decide what is living, what is just lifelike, and what is non-living. Because here in this example, it's clear what is the robot and what is the actor. In this case, half of the image is the head of Professor Hiroshi Ishiguro, but the second half is the head of his robot, and you cannot decide what part is the robot and what is the human being. And in similar way, if I show you the droplets and living cells, sometimes how you say what is the cell and what is the droplet and what is living and what is just lifelike, and when we can say it's completely non-living. So this we have to have in our mind when we think about life and artificial life. And back to the word robot, as I mentioned, we celebrated 100 years of the word robot last year. So we had the conference Artificial Life 2021 in Prague, but it was online conference. And this year we will have the A-Life conference in Trento. So if you are interested in artificial life field, you can join our community and attend the A-Life conferences. And also I edited the book with the title Robot 100. And the title Robot 100 was because it was 100 years after RUR, and also because 100 people contributed to my book. It was published in Czech at our University of Chemistry and Technology, Prague, but there will be the English edition, I hope next year, and it will be published by MIT Press under this title, Karel Čapek's RUR and the Vision of Artificial Life. And that's all from my side. And if you are interested in research on droplets or chemical robots and liquid robots and the Robot 100 book, you can check my webpage, or you can email me anytime, or even you can visit us, because this is the Campus Davice in Prague. And here is our building of the University of Chemistry and Technology in Prague. Thank you.
Amazing. Thank you so much, Dr. Čechová. It was absolutely fascinating. I'm really looking forward to this book. It really blew my mind to see the cells feel and move with nutrients. So yes, we'll come back to this at the question time. But as you were finishing your presentation on the definition of life, I feel like we have a very natural transition. So we've had the engineering perspective from Dr. Park, a chemical approach from Dr. Čechová, and now we're moving into more of a computer science approach with Dr. Sina Payne. So Dr. Sina Payne is an associate professor at the Sony Computer Science Labs, and she's a founding member of the Kyoto branch of this Sony Labs. So she's in Kyoto. It's 9 p.m. So thanks again for logging such a late time of night. I really appreciate. She's also a visiting researcher at Hokkaido University. And so she's researching AI and A-life and the different types of A-life, wet, hard, soft, artificial life. And she studies the fundamental characteristics of living systems and their computational properties. It's also to be noted one of her previous projects was in collaboration with NASA to predict life on other planets. So I'm going to hand over to you, Dr. Sina Payne.
Thank you for this introduction. I'm going to share my screen. Can you see my screen?
Yeah.
Okay, good. So I was thinking of giving a global introduction to artificial life, but Yitka did so brilliantly. So my talk will maybe be a bit short. So, oops. As Elsa said, I'm Sina Payne. I'm an associate researcher at Sony Computer Science Labs in Kyoto, and I work at the frontier between AI and artificial life, also called A-life. So I will give a very short introduction to A-life then, and I will talk a little bit about the links between A-life and AI. Then I will introduce a very important topic in A-life, which is open-ended evolution. And finally, I will take a bit of time to talk about my own research. So the beginning of my talk is again very similar to Yitka's talk. So when people hear artificial life for the first time, I think there are two reactions if you're not an A-life researcher. The first thing that people often imagine is this. So a kind of hybrid between robots and humans, and that would be artificial life. And the other type of people are people who are computer scientists, but not familiar with the A-life field. And when they hear artificial life, they mostly think about this famous game called the game of life, where you have little patterns of black and white cells, and these patterns self-organize using very simple rules. So A-life is actually, as a research field, in between these two things. We are not here at the hybrid stage, but we have long passed the game of life stage. So as Yitka said, artificial life, the motto of this field is life as it could be, which means that on one hand, we are looking for a working definition of life that would allow us to build life in an artificial system. And that might be surprising, but there is currently no definition of life that everyone agrees on. We don't have a good definition. And A-life researchers believe that if we have a good definition, we can build life. So the fact that we cannot build life means that we don't have a good definition yet. At the same time, we aim for more than just replicating the kind of life that we know on Earth. We want to be able to go beyond that to create new forms of life, maybe life that used to exist on Earth, so in the past, or maybe life that could exist in completely different environments, including other planets. So how can we have something that is both similar to real biological life, but at the same time different? This is the analogy that I use the most often. So here, for example, on the left side, you have a virus, biological virus. The function of this virus is to infect some organism and self-replicate and try to propagate as far as possible. Just below that, you have computer viruses, which also have the function of infecting something and then replicating and try to propagate as far as possible, like the biological virus, which is why they have the same name, virus. So they have the same function, but obviously they are built completely differently. There is nothing in common in the implementation of these two systems. You could say the same thing about DNA. So if you decide to define DNA as a way to transmit information between cells or between a parent and a baby, you could also have many different ways to transmit information. You could use anything, including bits in a computer to just transmit information between cells or between parent and offspring. So you can have things that have similar functions, but are implemented completely differently. So Yitka already talked about the 3 subfields of a life. I would like to add one subfield, which is art. So you have the 3 subfields: hardware, which is mostly robots; wetware, which is Yitka's research, and also everything that is biology and also physics, chemistry. And then you have software, which is more my field. So everything that is simulated organism, simulated systems. Oops. And so the last subfield of a life would be art. So a life asks very philosophical questions like, what is life? What is the difference between life and death, life and not alive? And so, of course, we have a lot of links with people who think about these questions, which are either philosophers or artists. I myself often collaborate with artists. This year we had performance, modern dance performance, where the dance troupe danced on the topic of artificial life. Here you also have different pictures of art projects that are not my project, but very interesting projects. Here you have an artificial plant. Here you have the android robot Alter, who is directing an opera. And then I want also to show 2 short videos. The first is this one. So this is very famous work by the artist Theo Jansen. These are machines that are very big. As you can see, this is the scale. And they are made of basically sticks and fabric. And there is no computer element. There is no electronics, no electricity at all. It's only mechanical. And yet these machines are able to work on different types of surfaces. They can also store energy. Some of them can fly. And all of this was built way before we had robots that could actually walk. When robots were still unable to go up the stairs or walk on sand or something, this person was already able to build these machines that have no real brain, no computer, but can have very biological looking motion like this. The other video that I want to show is actually more audio than video. It's an artistic piece by the group Alternative Machine, where they try to artificially reproduce the soundscape of the night. So if you go out at night and you're in the countryside, you might hear all kinds of insects and frogs singing, but they're not singing all over each other. It's very ordered. They have specific frequencies. They don't talk over each other. To reproduce these dynamics, but with simulated agents. The task of these agents were to communicate with agents of the same species while avoiding to be drowned out in the noise of all the other species. So let's see if I can find the place where it's actually playing, maybe here. Oh, more
the
here.
module
Maybe.
mainly in which it is installed.
So this is the kind of sound that you can hear in the background.
Depending on the environment in which it is installed, how the soundscape will evolve will change each time. The evolved sound, sometimes, is very similar to biological soundscape, consists of insects, birds, or frogs. Sometimes, the evolved sound seems more artificial sound.
So as you could hear, sometimes the sound is almost natural, and sometimes it's very artificial, something that you would not hear in a natural environment on Earth. So this A-Life field is very broad. So we have very broad open questions. The first big question in A-Life is, can we find a constructive definition of life, as I said? Can we find a definition that will tell us the difference between life, non-life, and death? And a definition that should also be not just for the life that we know on Earth, but for all kinds of life. The second question is the origins of life. So how do you go from non-life to life? What are the minimal conditions? The third big question would be open-ended evolution. I will talk about this later, but basically the question is, can you make a system that is evolving but never becomes boring, just stays interesting forever? And then you could add basically any questions you want, including, can we find life in the universe, etc. So I work more with AI and A-Life, and this is just my personal opinion about the relationship between AI and A-Life. I think AI is a function of A-Life because I think intelligence is a function of life. If you manage to build life, eventually you will have intelligence. And on the other side, it's very difficult to find an example of intelligence that is not related to life at all. So I think that AI is a part of the big field of A-Life, especially because A-Life is way broader than just intelligence. We're interested in any kind of learning, including organisms that are not humans, or even organisms that don't have a brain. And we're interested in more than learning, everything that is adaptation and evolution. You could even rephrase all of the big open questions in A-Life as being about AI. So instead of the definition of life, maybe we are looking for the definition of intelligence. What is intelligence? Can you define it besides just the intelligence that we know on Earth? The second question would be, what are the origins of intelligence? The third one would be, can we build open-ended learning instead of open-ended evolution? Can we find intelligence on Earth? So around us, we already have many different organisms that all have some kind of intelligence. But can we also find this intelligence in the universe? So now I would like to talk about the concept of open-endedness. I think most people come in contact with the concept of open-endedness through video games. When you have a game that is open-ended, the game doesn't have a fixed plot. You don't have to play step by step, do this, then do that, then you have the final boss. Open-ended games let you play how you want to play, and they let you make up your own rules. So open-endedness is characterized by the fact that something does not have an end, and the rules of the systems are always changing. Now, open-ended evolution is the big dream of A-Life researchers, because if we could build open-ended evolution in a simulation, so like a computer, we could basically, in theory, get everything from nothing. So what do I mean by that? It is said that Earth is a system that has open-ended evolution, because when you look at the history of life, you start with things that are not alive, and then you have life, single-celled organisms, and then you have these organisms, which are very simple, that evolve faster and faster to become more and more diverse, more and more complex, until you have a complete explosion of different species doing all kinds of different things. But so the important point in open-ended evolution is complexity that grows exponentially. That's not only true for biological systems, it's also true for technology. So here there is an image that takes the example of computers. If you take the first mechanical computer and the first electronic computer, there is a lot of time that was spent between these two inventions. But if you take the time between the first electronic computer and the first personal computer, for example, or the first personal computer and the first smartphone, the time between these inventions becomes shorter and shorter. So you have an exponential explosion of complexity of technology. And we want to obtain the same exponential explosion of complexity in a simulation. We want to find a system where you can start with almost nothing or a very simple organism, and then you apply evolution to it in your computer, and you get things that are more and more complex, even going to different types of animals or things that are intelligent or even maybe conscious. So you get almost everything from almost nothing. Now, nobody has actually managed to make such a simulations, and there's even a prize for under certain conditions. If you manage to build such a simulation, you could win $100,000 or even more than that, because really, if you can obtain everything from nothing, you can do whatever you want, basically. So now I will briefly talk about my own research. I have selected 3 projects that I work on. The first one is looking for life in the universe. The second one is the idea of replicating failures. And the last one is the evolution of cooking cultures. So the first project is looking for life in the universe. This is a project in collaboration with Caltech and NASA. And we're looking for a good way to say whether a planet could be a good candidate to have life on it or not. So at the same time, we don't want to use a traditional method, which would be, go to the planet and look for DNA, and if you find DNA, then there must be life. Because we don't actually know if life on other planets would have anything like DNA. We have no idea what life could look like. So we need a method that is very general, and this is what we propose in this paper. We basically take the light from planets and we analyze whether this, so we look at the planet for 100 days, and we take this data and we analyze the complexity of this time series, and then we compare the different complexities for different types of planets. So what we did is compare first Earth with Jupiter, and we found that Earth has a higher complexity than Jupiter, which is interesting because a few years ago there was this hypothesis that maybe there was some sign of life on Jupiter. But we found that Earth is way more complex than Jupiter using this method. And we also simulated some artificial planets. For example, a planet that has only deserts, a planet that has some continents and some water, but no clouds, a planet that has only clouds, and then a very complex planet with all kinds of surfaces. And again, we find that Earth is the most complex of this group of planets. The second project I want to talk about is the idea of failure replication. One thing that we often do in AI and often in life as well is to look for biological success. For example, humans are very good at recognizing images, or rather recognizing objects in images. So we think, okay, we will make an AI that is also very good at recognizing images. And when you do that, you're taking the risk that your artificial system actually works in a very different way than the biological system, because it's possible that these two systems by chance succeed at the same task, but actually have very different failure and success points. So if you want to avoid that, you should try to reproduce not only the successes, but also the failures to basically match the frontier between these two systems. So every time that the biological system fails, the artificial system should also fail. And if you do that, you can be more confident that your two systems are kind of matched, which give you a good argument to say that the two systems might work the same way, which is of course very important if you're trying to understand the biological system using artificial systems. This is something that I did for to study human perception. So I made a system that is able to generate new visual illusions. The idea being that a visual illusion is a kind of failure of your perceptual system. Your brain is telling you that something is there, but actually it's something completely different. For example, these images are static, but some people perceive some kind of motion. So we have now a system that is not only able to detect these kind of visual illusions, but also create new illusions. And from this, what we learned is that probably the reason why humans see illusions is because they are trying to predict the world around them. So if you build an AI that also tries to predict the world around it, it will become able to fail for illusions as well. The last project I want to talk about is Cuisine culture generator. Here, the idea is to try to replicate the history of one dish. For example, how did humans manage to invent bread, which is quite a complex piece of technology? Or how did humans manage to discover beer, for example? So you can use this simulator to try to find what kind of actions the first humans may have done to obtain these recipes. And you can also try to guess what was invented first. For example, was beer invented before bread or the opposite? You can also try to evolve entire cultures, so not just one dish, but several dishes that make basically the cooking culture of this specific people. And as a kind of opposite, you can also try to make up new histories. For example, what if tomatoes from America had never been introduced to Italy? So they would never have tomato sauce. What would Italian cuisine look like? So that's the goal of this simulator. Okay, that's the end of my talk. Normally here I would have a conclusion with a summary of what I talked about, but I tried to be very broad and talked about many different things, so there is no conclusion, except that I think that artificial life is a very interesting research field, and I hope I could convince you that we make some interesting research in this field. Thank you.
Thank you, Dr. Sina Payne. It was super interesting, in particular hearing your views on the relationship between intelligence and life, and yeah, it was very interesting to have sort of a broader view of what the concepts are beyond AI. Right, so I'm going to invite all the speakers to put their cameras on. As we're going to move to the Q&A session, we have about 20 minutes. We have some questions from the audience. So actually there was a question for you, Dr. Sina Payne, so maybe we can start with this question about the definition of life. From Fatima, she asked, I thought everyone agrees on the definition of life that basically says that life is a system that self-organizes and goes against the principle of increasing entropy. So could you comment on this definition of life and your own? And I imagine it could also be interesting to hear Dr. Českova's view on that afterwards.
Right. So I can already say that many people don't agree with this definition, although it sounds like a good, obvious definition. But actually, life does not go against the law of increase of entropy. If you have a living system, it tends to create a lot of entropy around it, while itself being more and more ordered. So actually, this law of thermodynamics is respected. So we cannot really use this definition. Of course, it's a definition that can be useful in some cases, but not everyone agrees on that. You could also say, if you were a biologist, maybe you would say, well, everyone agrees that, you know, life has DNA. If you have DNA, you have life. But of course, many people also disagree with that. And there are even cases where we don't know if something is alive or not, like viruses. Are they alive or not? People disagree on this question because viruses can self-replicate, but not by themselves. So if you have a virus that is just there, it will never change, like an object or a piece of dust. it has to be in contact with actually living organisms to kind of hijack this organism and be able to self-reproduce. So actually, yeah, nobody agrees on the definition of life.
Interesting. What about you, Dr. Chikova? What do you think?
I just fully agree with Lana that it is the general problem with the definitions, that there is no definition which is acceptable for everyone. So each field has its own definition. So as Lana said, biologists have different views than people from other fields. So yeah, but it's not just with the life, it's also with the intelligence, with consciousness, and with other terms that just, it depends who is creating the definition.
All right, and I guess it's a constantly evolving field of research. I'm going to move on to a question for Dr. Park. There was a question earlier for you, which is a bit left field. So the question was, is there any relation to microbial fuel cells, or is this completely different? And maybe I can add to this question or give a bit of context, because the way I understand microbial fuel cells, microbial cells are grown in order to produce energy. And in your case, we're talking about human cells rather than microbial cells. And they are also used to produce energy, but in this case, movement. And actually, I was wondering as well when I watched your presentation. So in the case of your biohybrid robots, the cells are used to contract and produce a certain type of movement. But could we imagine that at a wider scale, human heart cells could be used to create a sort of mega biological power station and be harnessed for their power and possibly fed with sugar or something?
Yeah, so for my biohybrid systems have to provide we have to provide the energy source like the glucose, and then they convert the glucose to the ATP. Mm-hmm. And then that is kind of so actually it's the biological material, biological cell, actually living cell actually doing that. So if we want to build a bigger like a force, then we have to integrating all of the tissue, like a bigger tissue form. But this is current technology really difficult to make the bigger size. Like right now we have only 1 centimeter size, but it's the current technology we cannot build the bigger size because of the several technical problem. So, but it's there's several research proposal we currently working on. So we want to build like 100 Newton or 1,000 Newton. So if your finger is a kind of motion, this kind of finger motion, it induced about 100 Newton force. Mm-hmm. So our first target is whether we can generate in this much of the force. That is our first target. But yeah, maybe that is not the right answer, but it's yeah, that's what we are doing.
Okay, amazing, thanks. And so can I ask as a follow-up perhaps, how do you see the future of your field and the potential applications of biorobots? And I guess that could also be a question for both you, Dr. Park, and Dr. Chikova. What could be the application of biological robots? Is the main advantage to be able to perform precision tasks, perhaps in the medical setting, or could you also tell us about the other applications that we could see in the future?
For me, it's the first application, obviously, to implantable the biological tissue. So it's for the surgical purpose, biomedical purpose. And then another one is one of the biggest the application is the culture's meat. So we want to build the large amount, massive production of the culture's meat using this kind of technique. And then another one is we also working on the NASA project. So we want to send to this kind of the artificial, the Biohazard System to send to the space. So we want to see that what is the response of this kind of human, like a tissue in the space. So that's another application. And yeah, that's kind of thing we trying to do. Yeah.
Great, thank you. Dr. Chekova, what's your views on the future applications of biorobots?
The thing is that, as I mentioned, I am artificial life researcher, and A-life is a very interesting field because the researchers are mainly curiosity driven, and sometimes the applications are like the byproduct of the research. These are the words of Hiroki Sayama, one of the artificial life researchers. So we are not driven too much with the applications. We are interested in the big questions. We study the systems because we do the basic research. So in my case, I am interested in the droplets, and each experiment that I do brings new interesting phenomena that I study. But in my case, it's not like that I have the objective to have the drug which will be good for all illnesses. It will help with cancer and with everything. In my case, it's different, that I look for new interesting behavior. But if we speak about the applications, so we can use the droplets, for example, for intelligent cleaning, as I have shown that the droplets are able to transport small objects. They can also transport the chemicals. So if we have some poison in the environment and we have the droplet, the liquid robot, which is able to sense this poison, it can bring some chemical which is able to neutralize this poison. So we don't need to distribute these chemicals for neutralization everywhere, but the chemotaxi can bring it to the place where it is needed. But I would like to mention, don't think about these liquid robots for human body. We will not use it for drug delivery, and because it's too big, and the chemical composition of my system is not good for drug delivery. Maybe for the skin, but maybe.
Okay, understood. Yeah, that's a fascinating example, the cleaning system, and I do understand you're working really on fundamental science. And as we're talking about fundamental science, I like to ask a question to Dr. Sina Payne about. It made me think, actually, both you and Dr. Park talked about the relationship between science and art. And obviously, most of the people in our audience are artists and designers. So you mentioned, Dr. Sina Payne, that you've worked with artists. You gave examples of artists, which was really amazing, how artists can contribute to the communicating on these concepts. And yeah, I was wondering what's your views on the role of artists and designers in advancing thinking or communicating science, or your views on the relationship between art and science?
It's a very interesting question, because when I was a PhD student, I always said I don't understand art, and I don't really like art because I don't understand it. But everyone around me was doing all kinds of different art. All of my colleagues, all of the other students were doing, amazing things with sound or images. And so my vision of art has completely changed thanks to A-Life research. And I think artists have different roles that they can play in the A-Life community research. for example, there is a very famous and big project in A-Life right now called Lenia, which was first introduced as an art project. So it had these beautiful shapes. I think I showed in my slide actually. You have some, it looks like cells that are dividing. Sometimes it looks like small creatures that are chasing each other. And at first it was presented as an art project at an A-Life conference, and it received the prize for best art. And then it became a research project. And now there are several teams around the world who are working on these kind of life forms that can even learn new tasks. So I think artists are both a source of inspiration for A-Life researchers, and also sometimes we are a source of inspiration for artists, of course. And in my case, in particular, when some artists want to collaborate with me, I have to explain my research to them, and they always ask the most interesting questions. So when I talk to scientists, they ask questions that I kind of expect. There is rarely a very new question. But when I talk to groups of artists, they always have this, I don't know, insights or like questions that come from nowhere. And sometimes I don't have an answer, or I have to think about it before answering.
Interesting. Well, fascinating to hear this art project that became a big research project. Coming back to you, Dr. Park, at the beginning of your presentation, you gave examples from science fiction films, and obviously in the world of design, design fiction is a big part of what design is. So I was wondering, same question, how do you perceive the relationship between science fiction and science, or between art and design, or design fiction and science?
Yeah, that is a really difficult question for me. By the way, my brother actually is graduate your school, so I'm figuring out that my brother is kind of the artist too. So when I communicate with my brother about this kind of presentation, he graduate major, his major was the fashion marketing, and then some of the biological kind of pattern can be used as a fashion design, the pattern, so coding pattern, or that is kind of the one of the thing. And also as you mentioned about the movie, so scientific fiction, and then it's definitely, I also kind of agree that in the future is the biohazard system is going to be surround us. So maybe maybe we also inspired by the science fiction too. So it's kind of we can provide some of the recent research to the artist, movie artist or something like that, and also movie maker. And also we also inspired. So we kind of inspired by the imaginary concept. So we can use their idea to build our biohazard system too. So it's kind of two way around. So yeah, that's kind of the thing what I imagine right now. So
thanks. Another question has come up for Dr. Chekova from Ravi. Can the rotating droplet be used for mixing, like a magnetic rotor mixer is used?
Yeah, maybe. I cannot say yes, I cannot say no. We have not tried it, but I think it could be possible. Yeah. And I would like to comment on the connection between art and science. So in my case, as you have seen the movies, they look very nice. And if I present it like the science, people ask like why it happens. You are the researchers, you have to be able to answer and explain why it happens. But if you are artist, you present something which is beautiful and people are just like, wow, it's art, it's nice. So I said that maybe I can switch to be the artist and to show these droplets like the art and don't care about why it is like this. And actually, because the experiments in the lab, you have a lot of good experiments, but also some experiments are with the problems. So I used the experiments that they failed, and I created short movie, which is titled Kapky, because it is in Czech, droplets. And I submitted this as the short movie to the documentary film festival Jihlava a few years ago, and it's available on my YouTube channel. And then also we created another short movie which is related to chemistry and beauty of the chemistry, and it is called Chemobronics, and it is related to chemical gardens. And also, if you are interested, you can watch it on YouTube in my YouTube channel.
Great, thank you. We're running out of time, but maybe I can take some of the quick questions in the chat. A quick question for Dr. Sina Payne was, could we get access to the Cuisine generator?
So not yet. First, because it's not ready yet. It's on GitHub, but it's private. And I want to make it really easy to use. Right now, it's just a bunch of code, and it's really hard to know what's going on. The output, as you saw, was just a black screen and some white lines on it. I want to make it a bit user-friendly before releasing it.
Okay, and there was another quick question for you. What's the name of the art project that turned into research?
So it's linear. I can type it maybe in the YouTube chat. Linear,
Yeah,
so
that would be
they
great,
can
thank
see it.
you.
Oh, done.
All right, it's 2:01, so I'm afraid we're going to have to come to an end. Sorry, we couldn't do more discussion and question. It's been absolutely mind-blowing to hear about your 3 fields of research. And yeah, I wish you all the best. And yeah, thank you for doing this fascinating work of advancing knowledge. It's really, really inspiring. Thank you to you 3.
Thank you. Thank you.
Thank you for the invitation.
Bye-bye.
Bye.
Have a good day or night.
You too. Bye-bye.